Hi everyone, thank you for watching this video tutorial on how to scan and grade Plasmodium blood smears with the ID-Fish Genus test kit. This video is going to go over the steps involved in reading and grading a Plasmodium fish smear that has been hybridized with the ID-Fish Plasmodium Genus test kit. To begin, you must ensure that you have the proper setup. For this we use a 100x oil immersion objective. You should make sure that you have an LED fluorescent unit with custom filters and that both of the power switches are turned on. You must also check to see that the slider is set to the blue filter. This will ensure that you will see a green excitation when looking through the eyepieces. When you are obtaining your samples, first ensure that they are no more than 2 years old. You can read a single smear slide or a double smear slide, whichever is more convenient. The smears should be made according to the instructions in the product insert included with the kit. The grading table for Plasmodium fish is as shown. It is the same grading scale that is used for Giemsa stain reading of thin smears. In order to call the smear negative, one must read a minimum of 300 fields and find no parasites. As seen here, a grading code of 1 would correspond to 1 to 2 parasites per 300 fields. A grading code of 2 would correspond to 1 to 9 parasites per 100 fields. A grading code of 3 would correspond to 1 to 9 parasites per 10 fields. A grading code of 4 would be 1 to 9 per field. And a grading code of 5 would correspond to an extremely high positive, having more than 9 parasites per field. Now we will go over how to efficiently and effectively scan the microscope smear in order to maximize accuracy. Since a minimum of 300 fields are required for negative and low samples, the best way to achieve this is to begin at one edge of the smear, reading down one column with the blue filter in place. You will be looking for green parasites. Then shift the field over in one direction and read up a different column. Repeat that 2 more times, and that should be adequate to get a sufficient reading. Since one column represents approximately 75 fields on a normal-sized thin smear, reading 4 columns would be equal to approximately 300 fields. Make sure to record the results on a form like this. If the slide is genus positive, you may optionally proceed to the ID-Fish PF/PV speciation test kit. Here are some examples of what to look for in a positive sample. I will denote true signals with green circles and artifacts or anything else indistinguishable with red circles. This is some footage of me scanning a PF control slide. You can see some nice examples of typical ring-shaped Plasmodium parasites inside of red blood cells. Their green color is indicating that they are Plasmodium genus positive. If you would like to further speciate these parasites, you can use the ID-Fish PF/PV combo test kit explained in a separate tutorial video on our website. Here is an example of what an artifact looks like. As you can see, the morphology does not look like Plasmodium, and a good way to double check that the signal is in fact an artifact is to look between 2 different filters, if that is available to you. For this test, we know that we are only looking for signals that excite in green, so anything that is also exciting in orange is definitely not a real signal. This is an example of what a real signal looks like. Here we are looking at a PF control slide, and as you can see, this donut-shaped parasite shows up well in green, and not in orange. That indicates that this signal is not an artifact, and that it is genus positive. Here is some scanning footage with some nice signals that I will circle in green. I also want to show you an example of what a messy slide with a lot of artifacts looks like. I am circling them in red. If you encounter a slide like this, or a slide with similar problems, it is best to refish the sample. There is also another video tutorial called Plasmodium Troubleshooting that may help you address some other problems you could run into. This is a zoomed-in picture of what a gametocyte looks like with fish. This classic banana shape is a good clue that this genus-positive slide is likely Plasmodium falciparum. Here is another picture. This is a genus-positive signal on a Plasmodium falciparum slide. Okay, well that wraps up this video tutorial. I hope it's been helpful. Please view our other tutorials for examples of artifacts and tips on troubleshooting. If you happen to have time, there is also a brief 10-question customer feedback survey on our webpage that we would appreciate if you completed. The feedback helps us very much in figuring out how to serve your needs better. Thank you for watching!